our products and services mostly are related to life sciences. Recently, we have jointly committed to a mission. ACI and Genofax. Recently, we have gone to a mission where we agreed to jointly share the glorious joys, the failures, the sadness, and the successes of life sciences. And it's allied fields. And we mark that mission with this small gathering. I must say, to me, it's a precious, I know that you'll agree with me, it's a precious gathering. And we call it Celebrating Science. We are very lucky, we are blessed that we have two prestigious scientists, both from Australia, Dr. Mark Tester and Dr. Abed Chaudhury. Today they will share their journey with applied life sciences to make this a better world. To make this world a better place every day. We are incredibly pleased to have Professor Zeba Swaraj, the eminent rice scientist and champion of salt-tolerant crop in Bangladesh, to introduce today's speakers to you all. Dear all, let us welcome these celebrities with a round of applause. We thank you again for your time and gracious presence. Thank you. Good afternoon, everybody. I add my thanks for all of you to come here. On a Friday afternoon, so we usually eat more lunch. We have a heavy stomach and it's rice, it makes us sleepy, but despite that, everybody is here. So it's my great privilege and honor to introduce Dr. Mark Tester, who is foremost someone who loves plants and is excited by science. A scientist par excellence who brings a unique blend of passion, keen interest, and excellence in biological science. He loves understanding how plants work, especially in tough conditions where it is hot, dry, and salty. He is a professor at KAUST, which is King Abdulaziz University of Science and Technology, and has also worked at the Universities of Adelaide and Cambridge. He co-founded Red Sea LLC, which is a company, and he is now enjoying developing saltwater-based agricultural systems to improve the sustainability of food production. Furthermore, he has devoted a lot of his time for the use of science in improving lives and the environment for a sustainable future. His talk will tell us about his major discoveries and its impact in the Kingdom of Saudi Arabia as well as in Bangladesh. And this morning he visited my lab and he loves students as well, so that was a new discovery. So now I'd like to introduce Dr. Abed Chaudhury. Sorry, I'll just do it at once. Sorry. So Dr. Abed Chaudhury, he needs no introduction in Bangladesh. He is a very well-known and respected geneticist based in Australia and Bangladesh. He became famous for discovering the apomixis genes, which allow reproduction without hybridization. Much research has followed after this discovery, which is a key to multiplication of high-performance hybrids without segregation. Currently, he has been in the news for Panchobrihi rice, or rice which flowers 5 times. He has carried out important work in agricultural sciences and has written books on science in both Bengali and English. For many many years now, he has been working relentlessly and passionately to convert his home place, Kanihati in Sylhet, into a rural hub of intellectual activity, particularly in educational improvement and reform. Both of them will be speaking to you one after the other. So thank you very much. We'll see a short video on Dr. Abed Chaudhury and his amazing story of Panchobrihi rats.
বাংলাদেশের অর্থনীতিতে বড় ভূমিকা রাখে কৃষক। কৃষি নির্ভর বাংলাদেশের অর্থনীতিতে বড় ভূমিকা রাখে কৃষকের উৎপাদিত ধান। বিপুল জনসংখ্যার এই দেশে মানুষের মুখে অন্ন তুলে দিতে উৎপাদন বৃদ্ধির কোনো বিকল্প নেই। তাই ধানের উৎপাদন বৃদ্ধিতে নিরন্তর গবেষণা করে চলেছেন বিজ্ঞানীরা। তেমনই একজন বিজ্ঞানী মৌলভীবাজার জেলার কানিহাটি গ্রামের সন্তান আবেদ চৌধুরী। তিনি আবিষ্কার করেছেন পঞ্চবিঘি ধান, যেটা বছরে 5 বার ফলন দেয়। প্রথম রোপণের 110 দিন পর ধান কাটে, তারপর 45 থেকে 50 দিন পর ধান কাটা যায়। আমি ভাবলাম যে ধানটা একবার ফসল দিয়ে মারা যায়, এটা কেন হবে? এটা কি 5-10 ফসল দিতে পারে? তো এই হাইব্রিড তো একটা একবার ফসল দিয়ে কৃষক হাইব্রিড বীজ কিনে একটা ফসল দিয়ে শেষ হয়ে যায়। আমার এটা 5 বার দিই। পঞ্চবিঘি ধানে উৎপাদন বেশি, খরচ কম। এতে কৃষকের সময় যেমন বাঁচবে, তেমনি সাশ্রয় হবে অর্থ। খাদ্য সংকট মোকাবেলায় পঞ্চবিঘি দুই ভাবে অবদান। একটা হচ্ছে যেটা উৎপাদন বাড়িয়ে দেবে, 5 গুণ বাড়াবে না 3 গুণ বাড়াবে না 2 গুণ বাড়াবে, এটা বিভিন্ন দেশের পরিবেশ সবকিছুর উপর নির্ভর করবে। তাই বলা যেতে পারে মোটা দাগে যে সিগনিফিকেন্ট উৎপাদন বাড়িয়ে দেবে, এটা একটা গেম চেঞ্জার। পঞ্চবিঘি ধানের শিকড় অনেক দীর্ঘ হয়, তাই এটা প্রাকৃতিক দুর্যোগে কম ক্ষতিগ্রস্ত হয় এবং মাটিতে কার্বন শোষণ বাড়িয়ে দেয়। ধানগুলো অনেক দীর্ঘ এবং বিশাল শিকড় তৈরি করে পঞ্চবিঘি মেথডের কারণে। তো সেটা আবার কার্বন সেকুয়েস্ট্রেশন, মাটিতে কার্বন বাড়িয়ে দেয়। যেখানে 1% এর নিচে কার্বন, সেখানে এটা 3%, 2% এর উপরে হবে। প্রকৃতিতে ঘাস জাতীয় কিছু কিছু উদ্ভিদ রয়েছে যেগুলো পরাগায়ন ছাড়াই বীজ উৎপাদন করতে পারে। উদ্ভিদ বিজ্ঞানের ভাষায় একে বলে অ্যাপোমিক্সিস। ডক্টর আবেদ চৌধুরী ধানের ক্ষেত্রে এই অ্যাপোমিক্সিসের প্রয়োগ করে কৃত্রিম বীজ উৎপাদন করতে সক্ষম হয়েছেন। ক্রম করা যেতে পারে দুই ভাবে। একটা হচ্ছে যে বীজটাই ক্রম হয়ে যাবে, যে পদ্ধতির নাম হলো অ্যাপোমিক্সিস। আমার প্রথমে চিন্তা ছিল যে ধানকে আমি ভেজিটেটিভ করব, এটা বীজের মাধ্যমে জেনারেশন টু জেনারেশন যাবে না, বীজটা আমরা খাবো, চাল বানিয়ে খাবো। এটা ছড়িয়ে দেওয়ার যে প্র্যাকটিস, যেটা আমাদের নেটিভ গাছ, আপনি জানেন যে গাছগুলো কোনো বীজ নাই, কেটে 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 মানুষ লাগিয়ে। তো আমার স্বপ্ন ছিল ধানকে আমি এখন করবো। জলবায়ু পরিবর্তনের ফলে পৃথিবীর তাপমাত্রা বাড়ছে। পরাগায়নের সময় যদি তাপমাত্রা 39 ডিগ্রি সেলসিয়াসের বেশি হয়, তাহলে ধানের বরাগুলো নষ্ট হয়ে যায়, ফলে ধানের বরাগায়ন হয় না। এতে ধানের জিটা হয়, ফলন কমে যায়। কৃত্রিম বীজে এই সম্ভাবনা নেই।
আমি যে কাজটা করলাম যে শুধুমাত্র ফিমেল এর থেকে কিটা প্রাণী হতে পারে কিনা, কোন পুরুষের কন্ট্রিবিউশন ছাড়া। এই প্রশ্নটা আমি করেছিলাম 90-এর দশকে, এবং দেখিয়েছিলাম যে একটা ফিমেল এর থেকে এটা প্লান্টের ক্ষেত্রে, মানে বৃক্ষের ক্ষেত্রে, যে ফিমেল এর থেকে এটা হওয়া সম্ভব, এবং মেইলের রোলটা একটু কত বেশি ইম্পর্টেন্ট। এটা আমার মেজর ডিসকভারি ছিল।
আমাদের দেশে ধানের 3টা মৌসুম, পঞ্চবিধি ধান 3 মৌসুমে সমান ফল দেবে। কিভাবে তিনি এই অসফল সাধন করলেন?
এটা প্রথমেই আমি এটাকে আড়ি করে দিয়েছি পঞ্চবিধি করার আগে, যেটা আড়ি হাতে। এখানে যে সিজনাল যে ব্যারিয়ার যেগুলো আছে অনেকগুলো, যেমন অনেকগুলো আমন আছে, আমন ধান যেগুলো, সেগুলো তো গ্রোথে ফ্লাওয়ারের জন্য একটা দেয়ালেন্সে একটা সার্টিন দেয়ালে নাইট পায়, তখন ফ্লাওয়ার। এই জন্য আমনের ধান আমনেই শুধুমাত্র হেমন্তকালের সময় ফ্লাওয়ার করে। মেকানিজম যেটা, ভিতরে একটা ঘড়ি আছে, ওই ঘড়ি দিয়ে তারা মাপে যে ওয়েদারটা কেমন, দেয়ালেন্স কেমন, তখন ফ্লাওয়ার করে। ওই মেশিনটাকে ভেঙে দিচ্ছি, বলতে পারে।
গবাদি পশুর অন্তর থেকে মিথেন নির্গমন হ্রাস ও মাটিতে কার্বন আবদ্ধ করার পদ্ধতি নিয়ে আবেদ চৌধুরীর একাধিক পেটেন্ট রয়েছে। এছাড়াও তিনি 2022 সাল থেকে জেনোফ্যাক্স নামক স্বাস্থ্য বিষয়ক প্রতিষ্ঠানের সহ প্রতিষ্ঠাতা ও প্রধান বিজ্ঞানী হিসেবে দায়িত্ব পালন করছেন, যা অস্ট্রেলিয়া, বাংলাদেশ সহ বিভিন্ন দেশে কার্যকর হয়েছে।
And after the sequencing, we get to know their identity, we get to know their relative abundance.
ডক্টর আবেদ চৌধুরী রঙিন ভুট্টার ক্লোন উদ্ভাবন করেছেন। তার উদ্ভাবিত রঙিন ভুট্টা বছরে 4 বার চাষ করা সম্ভব। জেনেটিক্যালি মডিফাই করে তিনি এই ভুট্টা তৈরি করেছেন।
রঙিন ভুট্টা ঠিক আমার আবিষ্কারিত আমি বলবো না। রঙিন ভুট্টা তো সাউথ আমেরিকাতে আছে, সেন্ট্রাল আমেরিকাতে আছে। আমি খুব পাওয়ারফুল এবং মাল্টি কালার, যে মধ্যে যত কালার আছে সব কালারের হাই ইন্ডিং ভুট্টা আমি তৈরি করি।
ডক্টর আবেদ চৌধুরী পঞ্চদিঘি ধান নিয়ে গবেষণা অব্যাহত রেখেছেন।
আমার যে অ্যাকচুয়াল গোল যেটা, ধানকে সম্পূর্ণ ভেজিটেটিভ করে দেওয়া, পেরেনিয়াল করে দেওয়া। আপনি সুগার কেন দেখেন বা লেপিয়ার গাছ, একটা আমরা এক্সাম্পল, এটা প্রোগ্রেশন অনেক সহজ। তো আমাদের ধান যদি এরকম হয়ে যায়, অন্তত কিছু ভ্যারাইটি আমি করে ফেলতে পারি, তো এটা তো ফুড সেক্টরে একটা বিরাট অবদান।
পৃথিবীর বিপুল সংখ্যক দরিদ্র মানুষের প্রধান খাদ্য ভাত। ডক্টর আবেদ চৌধুরীর এই আবিষ্কার পৃথিবীর সর্বপ্রতি মানুষের মুখে খাবার তুলে দিতে বিপুল অবদান রাখবে।
I invite Abed Chaudhury, Dr. Abed Chaudhury to deliver his talk.
I think we should go straight to the slides. I think the perennial rice part was covered already by the documentary, so I will not go too much into it, except to say that what has been achieved. So we were able to, first of all, in the journey of apomixis, we started by getting what we call fertilization independent seed. That was done in the 1990s. I discovered the genes involved in the fertilization independent seed pathway, so that no pollination is required. Those genes were obtained in Arabidopsis initially. Then we used the heterology of these genes and got the cognate similar genes from rice. And then in a 2-gene knockout CRISPR activity, we were able to get fertilization independent seed in rice. And that was covered, published last year in the cover story in Nature Plants. And now we are trying to combine it with the apomyosis gene, the so-called MIME, and instead of meiosis, you have mitosis. So other people published that. We are combining that and to get functional apomixis using our FIS system. And that work is ongoing in Australia, and hopefully the non-GMO part will be done in Bangladesh when we select for modified genes to make everything good. So what will be the outcome? Outcome will be that when you make a very good hybrid, and the best hybrid are made by ACI, I'm told. So that hybrid, when the farmer gets the seed from the hybrid, it segregates away the trait, and you have to come back to buy the seed again. However, when we kick in the apomictic system that we are developing, then the hybrid seed will, the babies of the hybrid seed will also look like hybrid seed, and there will be clonal propagation of hybrid vigor to hopefully through eternity. So that is the goal. In reality, I'm sure it will break down and all kinds of things will happen. So there are two ways to make clonal propagation of any crop. One is the clonal propagation by a seed, where you suppress all meiosis, and the seed is a mitotic replica of the mother plant, and that is by apomixis. So a lot of crops have apomixis, and we created synthetic apomixis. I created synthetic apomixis from the scratch, step by cross-cutting, fertilizing, pregnancy, etc. And then we have functional apomixis, so we can deploy it. The second way is to do by vegetative budding, the good old system. Like you go to a tea plantation, there is no seed. The tea plantation propagation is by just vegetative budding. Right? And all the Napier grass is in the grass family. I grow Napier grass just to get inspiration from it, so that I know what rice should look like. So Napier grass does very well, and it's a very viciously weed-like character. So imagine making rice into Napier grass. I mean, you have to imagine it first to make something happen. So that is doable. That is not like there is no divine law which says that rice will never become like Napier grass. It will. And in that journey, we have made rice 5 harvests. We have not yet made it like Napier grass, but we have created 200 independent 5 harvest rice in separate genetic events. So I'm hoping when I put in all the combinatorial, even with traditional Mendelian genetics, when I find one with the other, I'm hoping, and spray and pray kind of way, I wait that maybe it will become totally familiar. We don't know, but that journey has become. So that is the sum total of it. And in addition, I became part of an Australian company called Bio, and we found a way of taking carbon dioxide back and storing it into soil. And with my New South Wales colleagues, we raised a lot of money. Very big people like the owner of Google joined us, and so that company is doing very well. It's called Bio. And I myself had a solitary discovery where I found that some soil fungi can make an enzyme called vanadium haloperoxidase, and that removes the cattle rumen entirely. So that was also very successful with the collaboration with CSIRO. And now a new company is being spun out, probably announced next week, called ROM, R-O-M, from Roman, and kind of rhyming with long. So that is also my work of me and my close colleagues, which will come up. So this is the kind of status report of it. However, I would like to, I'm very happy to be here. First of all, I want to really thank Professor Zawasiraj and my close colleague, Montas Faruqi. I'm very happy to be speaking together with Mark Tester. Mark is coming here. This is his second year here. Last year he came and he traveled all over the country, went to the south with Professor Zawa, came to my village, and he's back again this year. So we must be doing something right. I think I'm feeling very positive because Mark is a very he's a guy who doesn't go everywhere, right? So the fact that he keeps coming back and say something about it. So I very quickly now, because we want to give Mark a lot of time to talk, so I will not go on and on, but I want to say a few words about Genofax and the science of Genofax. So that's what I will say, and then give the podium to Mark. So the first slide. So what happened? This is a kind of a popular science magazine cover story that just came out like last month. And this shows something very important. It shows the integral connection between the brain and the gut, which people were saying gut-brain axis, and this word has been around. In ancient Bangla literature, we say, "Jantam na tomar pete pete to buddhi." প্রাচীন বাঙালিরা, just relax for a second. বাঙালিরা জেনেছিল যে পেটের মধ্যে বুদ্ধি থাকতে পারে। কিভাবে তারা কি গাট মাইক্রোবায়োমের কথা জানতো? আমার মনে হয় এটা একটা metaphor. It's a poetic thing that you have so much brain in your gut. Literally, there's an old Bengali adage which says, the mother saying to the daughter when he's been in distress, that you have so much brain, in fact, most of your brain is in gut. This is why you're so mischievous, right? So it turns out to be literally true that a lot of our brain neurons are actually in the gut. We knew this for a long time. And now it seems like a massive number of what we call brain function are in fact being controlled from gut. And how is it being controlled from gut? We go to the next slide. And that is kind of the story of the genesis of Genofax as a company to find out, so to speak, that how the gut and the brain is connected. And I came up with the idea that it can be best understood with the doctrine of 3 books of life. So what are the 3 books of life? Next slide. So the first book we all know, Arthur and Crick discovered it, is the genome, which is the combination of all the DNA in our chromosomes, known as genome, which is a great system of saying "om" after to indicate a combination, grand combination. So the genome is what I call the library book of life. It's a massive, voluminous, multi-component 23 chromosome, and I want to call it complete and also silent, by and large silent. Then the second one is what we call the epigenome. Can we move this flat, please? Technical issue? Okay. So the second one is the epigenome, which is what I call the audio book. There only a small portion of the whole chromosome is expressed at a time. So imagine you have the whole physical body of the whole genome in every cell. Every cell has all the DNA needed for the whole organism. Yet the cell becomes a brain cell or a skin cell or a hair cell. How does it do that? It has all the information. Why is it hair as opposed to brain? So this is a big question in developmental biology, that how does the cell differentiation, they call it, happen? So the skin cell is completely different from the brain cell, but each one of them have all the DNA in their body. So to make it very clear in a popular science way, I said can be expressed by what we call audio book of life. So imagine this grand book, and only page number 7, 13, and 51 is made expressed through audio, and therefore you become skin cell. And from the same book, page 53 and 87 and 94 is made audio, and then you become a brain cell, and so on and so on. So from the same code, you can create multiplicity of function by keeping everything silent except expressing a subset of the cell. So this is basically the whole story of epigenetics. And the way you do it is very interesting, actually, if you want to think in a very simple-minded way. Because the code is immensely large, and the code is wrapped in nucleosomes, and they're all massively wrapped structure. So out of the wrapping, certain things become accessible, and that determines certain functionality, that determines whether cancer will happen or not. And that is being controlled not by the code, the genetic code, but the epicode, you can call it epigenome, which is roughly speaking how it is packaged, as well as methylation and small RNA, etc. And finally, about 20 years ago, when DNA sequencing became extremely easy and cheap, we got access to the third book of life, which is the microbiome, about which we knew nothing even 40, 50 years ago. So we used to think that yes, we have in our body, in our bacteria, but let's kill the bacteria with the data or antibiotic. That is how Biodiversity taught us, and this was our microbiology, that we must destroy our bacteria. And all the bacteriological department thinking and research was how to destroy all the bacteria, because bacteria by definition is bad. We also, there's an award of what's called germ. So you have to destroy your germ. But then when DNA sequencing started, because these germs can never be isolated, we found that our whole body is full of germ or microbiome. In fact, they have more cells than our own cells. And we are carrying it in every nutrient body of our body, but the big bulk of them are in our gut, and therefore gut microbiome. So this is the third dark book of life and the most recent book of life, and that can only be read by 16S, which is kind of a flag every bacteria has to identify itself. And you sequence the whole material from your poo. So you go to poo, and then you can send this off, and you can isolate DNA from your poo, and then do 16S sequencing, which identifies every bacteria and millions of them. And then you are given some characters to which bacteria you are carrying and what number, and that is extremely meaningful. That is predictive of what disease you're going to have. It is predictive of whether you have premature aging or whether you have early onset diabetes, etc., etc. The whole field is emerging. Every day there's a new discovery in it. And I met Jahangir Alam, who is the co-founder of Genofax, in a social gathering like this in Sydney, and I said, why don't you do this? Because you are spending your time doing electrical engineering and biology. So out of this great discussion, Genofax emerged. Next slide. And around the same time that I was talking to Jahangir, this Savante Babu got Nobel Prize because he found all the archaic DNA from ancient skulls, and he found that there were genes and which people were involved, but they found this whole field called nucleogenomics, where it looks like certain genomic single nucleotide polymorphism we had in our ancient DNA determine what kind of disease at the end we have. Next slide. So that a core lesson is that we mated in ancient times with another A-type hominids called Neanderthal or Denisovans in Asia. And out of the collision of these two genomes, the Homo sapiens genome and the Neanderthal and the Denisovan gene, out of the collision, a lot of SNPs emerged by a process called hybridized genesis and set in motion the process which gave us the modern human race. And when we look into what disease we are prone to, what disease we are getting, or how to predict disease, we have this great evolutionary theory of Neanderthal. Next slide.
The streaming shows that you will face so
I'm getting younger. So there are all these numbers that 272 million people globally are on the moon, and we have always been on the moon. So my ancestors came from Yemen, 10% of my DNA is Chinese. So it's all after DNA sequencing we know this, that we are not from one place, and we are connected in Bangladesh by language. Ethnically, we are not the same people. That's it. Next slide. So this shows the grand movement, recent movement, our own movement, but also the movement of the human race. And why the movement is important? Because if you are from Central Asia, let's say, as opposed to China, so 10% of my SNPs are of Han-Chinese origin. So I am prone to certain diseases, which maybe my first cousin is not prone. So this is as heterogeneous as that. And we have to be aware of that, and we can only find out by sequencing. So ACI is a very forward-looking organization. We had very good discussion with Dr. Arifudullah about having a very high-powered, competent, and kind of a modern, informed genetic sequencing facility. Then hopefully we'll take steps. Dr. Ansari is here. So we will want to partner with Genofax and ACI, hopefully in future days will partner to look into this issue a bit more. Next slide. So this, I will very quickly run through this. So most of the drugs that you take are given to you based on studies that were done in the European population. because European population has been studied, Asian population has not been studied. So as a result, we need to change it, because this is not like democratizing therapeutics. Therapeutics is very undemocratic. So maybe a study has been done in Scandinavia, and then out of that study came how much drug should be given. So now a patient goes from Burkina Faso or Bangladesh goes to the doctor. He's given the medicine on the basis of the European study. So this medicine is called one size fits all medicine, which is the current state of it. On the other hand, if my DNA was sequenced, even S&P level, not the whole thing, then a much better therapeutic protocol will emerge, and that's what we want to do. We want to kind of democratize the healthcare genetically, so to speak. Next slide. So we have several goals from Genofax, and I don't want to just go into the whole list, but we basically want to create in Bangladesh a new healthcare system, a new diagnostic system, a new therapeutic intervention system. And for that, from Genofax, which is a startup company based in Sydney and a little bit in San Francisco. But we want to partner with mega organizations such as ACI in order to go into this journey. And hopefully this seminar is a kind of a springboard for that kind of achievement. Next slide. So we have a test kit, which later I think you talk to my colleague, Mohammed Salaki. We have already in business in Bangladesh. So you can click on your smartphone, download an app, and the test kit will go to you, and you give your biological sample. It goes away to the sequence, and out of the sequencing, an advisory will be given to you about your lifestyle, what you should be eating, where you are heading. So you might be metabolically, you may be biologically only 18 years old, but your metabolic health might have 40, because you have done so many bad things to yourself. So this sequencing will give you the metabolic health. Another person biologically 60 plus might be metabolically healthy. So these concepts were not even there before, 30, 40 years, and it has emerged now. Next slide. So this is a new part of my story. I'll stop here and pass it on to Professor Mark Stell, and I think collectively we'll answer questions in the general. That's the plan, right? Thank you, Mark.
Right. So, quickly technical. When do you want me to stand, or am I allowed to walk? Can I walk, or do I have to stand? Am I allowed to walk?
Yes.
Okay. I'm just checking with the technical guys online.
You're allowed to.
Because naturally I walk. So. All right. Look, I'm pretty amazed. I have to admit, this is a very impressive gathering with some very senior people in this very big company, and so many scientists and many others here. So thank you for the invitation, and thank you all for coming. It is a great honor to be here. And I mean, what I'm doing is I'm a plant scientist, and in some ways I'm a fairly simple plant scientist. I'm interested in plants, as Ziva said earlier, and I want to understand how plants work, and then see if we can use that knowledge to try to improve plants. And in the last 10 years or so, I've really taken this to the next step, and not only want to try to improve plants, I really want to deliver that research as well. And I've become increasingly passionate about this as I travel more and I see the state of the planet, and I really see how we need to be delivering. The planet really, really needs this. And so I now talk to university presidents and government ministers and so on, and try to help provide an environment in which us academics are actually delivering, not just talking about it. for today. What I'll do today is try to tell you a little story about some of the plants that we have been developing and delivering. And it's great to have Abed's talk because, I mean, he's a real geneticist, a real molecular biologist. I'm a plant physiologist, but I've been coming into the genetics field increasingly, and I'd like to, it's really good to have you in the audience because some of the work that we've been doing is different to yours, but it's got surprising similarities about trying to develop seeds that are more powerful and more easily reproduced, more cheaply reproduced for use by farmers in open fields, especially in open fields. Okay, if you go to the next slide, this just starts to, what I've done is I didn't realize I was going to be giving you a talk like this. I thought it was going to be a smaller, more informal chat. So I've taken a talk which I prepared to give on Sunday at the University of Dhaka at their institute that helps encourage entrepreneurial activity. So some of the slides towards the end are not relevant, but there's a little academic context, should I say, which is just, we need lots of water for food, and the surprising amount of that food is produced by irrigation, and about half of the water for irrigation comes from groundwater, and every aquifer that is being used for irrigation is being depleted. So a large fraction of the food that is being produced on the planet is actually being produced seriously unsustainably. And I don't just mean some tree hugging way. We're talking about we're using groundwater and depleting groundwater reserves, so we have to change somehow. So this is the context I come from, and there's heaps of water on the planet. It's just that most of it, by far and away, most of it is salty. And so if I'm able to unlock that salty water, the seawater and the brackish groundwater, and use this for food production, that can be really quite a significant contribution to the sustainability of food production. So we're going to unlock something, we need a key. And for me, we're living in fantastic times. A lot of the people in this room are a bit older. I do like talking to students, you're right to see that. But I mean, we're living in fantastic times now in plant science, in biology generally. You know, like Abed's talk where he's going to sequence 3 different books and do this as a service. That's correct. It's the same in plant science. We've got remarkable abilities to cheaply, accessibly sequence genomes, sequence epigenomes, and so on, and use that knowledge to try to accelerate, turbocharge plant breeding and crop improvement. And when you put the genomics revolution hand in glove with this 4th industrial revolution, where we've got the AI, we've got the computer vision. People forget about computer vision, it's really, really powerful, and so on, the IoT, sensors. If we put this together, then we can also be measuring features of the environment and of the plants themselves in that environment. Put that together with the genomics, you really start to get some powerful crop improvement possibilities. So I think we've got opportunities to have serious innovations in crop improvement. And we can apply this in 2 different ways. We can get the crops that we've got and make them better, and that's fine. We've been doing that for a few thousand years, so we can do it now faster and better and more powerfully. We can also get plants which aren't crops, which have got remarkable properties, and turn them into crops. And so we can go in both directions. And I think both of these directions have got opportunities. Now, at a company like ACI, you've got shorter time frames, greater risk aversion, and so on, which is perfectly respect. So often these approaches can be and should be done in universities, and then we need to be transferring that information and knowledge from the universities into the companies. This is something I want to discuss over the next couple of slides, because we've really got a lot of problems in that space. Okay, so this is what I'm going to the students and the academics. You start your ground applications with, oh, we've got to save the world, and we've got all this water running out, and so on. And we can talk about this stuff, but are we actually really doing it as academics? On the next slide, what you can do is start to look at the papers which are being published with genes being characterized and do all sorts of miracle things. Hundreds, thousands of papers being published. And how many of those genes, how many of those discoveries are really really being delivered. All right, maybe Abed's rice, that sounds pretty amazing. That could be delivered. One or two others maybe, BT, great, amazing, fabulous, breakthroughs. But it's a handful, come on, out of those thousands. So I'm really sick of this. I'm sick of watching my academic colleagues talking, publishing their papers, getting their awards for that, and not actually transferring the delivery. So that's where I'm trying to get very, very serious. Next slide. This is just the same story, only for salinity that I published many years ago. So I call this, we talk about startups going into a valley of death. We have all sorts of valleys of death of various sorts. And I think there's a big, grand valley of death between the academic research, with all of that being very well established, and companies like ACI and many companies which are delivering improved varieties. And there's this huge problem in the middle where there is not enough transfer of knowledge, information, discoveries, innovations from the academic to the applied space. And so that's where I'm getting very involved. Right, let's move on and show you an example of, keep going. Oh, that was a paper led by Ed Buckley who I was telling you about it. Go on, that's for academics. Go on, next slide. Right, okay, so this is just one example which actually happens to, which involves Bangladesh, which is work which I started way back in the early 2000s, over 25 years ago, where we were working in Arabidopsis, again like Abed was for Apomixis, and we were studying the single gene that we knew the properties of. It was transporting sodium, and if you express this gene in the middle part of the plant adjacent to the xylem vessels, it can remove salt from the transpiration stream before it gets to the shoot. So a pretty good gene. And if we overexpress this gene specifically in those cell types, so we had to develop the tools to do this back then. So we did that, then we overexpress the cells, you get big increases in salinity tolerance. This is in a silly little model plant that's of no relevance to anything. But what then happened was at that time we were doing this work, an Australian wheat scientist who was working on salinity, called Rana Mums, walked into my laboratory in Cambridge and just said, I've got these 2 bags of seed, and one's got 1/5 of the sodium in the shoot than the other bag of seeds, from the other bag of seeds. What do I do now? Why is this the case? So I thought this was probably a lifetime's research, and I could have a PhD student, another PhD, sit there in Cambridge thinking I'm smart and everything's good. One PhD student now, in this age of genomics, did this work in 3 years, identified the gene, identified the mechanism, was then able to apply it to wheat. Rana Mums and CSIRO in Australia, Abed's institution, then integrated this into Australian commercial varieties of wheat. We did the field trials, and what we found was that the gene did nothing in a lot of the soils. All right, it didn't have a yield penalty, which is good. It didn't have any benefits either. But if you go to low yielding fields, which have got high salinity, you get in the order of a 25% yield increase. Amazing. Headline grabbing results. It was in New York Times, everything. We were on the front cover of Nature Biotechnology. Everybody was happy. We were happy. Shot off to the Australian wheat breeders, and they go, not interested. What do we care about this? And then you sort of run away back into your hole and go, those people are idiots. And then you actually think about it a bit more. They're not idiots. They're responding absolutely correctly as business people who are getting their rewards, their profits, from increased yields and increased yields in the field, because it's an endpoint royalty scheme in Australia. You don't sell seeds, you get the money from the amount of seeds that have been harvested from the field. They were obviously targeting the high yield fields, all right, because there's more tonnage coming out. And if you do your sums, if we say it's a 15% yield increase, over 34% of the fields of Australia that are significantly affected by salinity, you're only contributing about 2.5% yield increase to the national yield, to the national harvest. forget it, all right? That's 2 years of wheat breeding. Let's just keep accelerating our wheat breeding as normal wheat breeders. That was their thinking, and they're correct, all right? My problem is that I'm thinking about that 34% of the land where we can have 15% yield increase, all right? That's 34% of the farmers. That's a lot of people. And so, in many ways, if we look at that valley of death, there's this disconnect between an academic's thinking and about thinking of, government policy, private industry. It doesn't matter if it's public or private in this case. You're looking at the national yield, 2.5%. I'm thinking about that 34% of the farmers. And so, in many ways, it's the responsibility of academics in universities and not-for-profit institutions to do the delivery, not for the companies and the governments to do those deliveries. So that's one thinking which was pretty was a big learning for me about how you can get really spectacular academic results based on some damn good science, and you don't get anywhere still because you're not really thinking about your market and about the real impact at the level of the whole society. So I left Australia. Rana Mum's retired. We thought things were sort of over, but Rana Mum's technician kept working with Richard James. And then he started collaborating with scientists here in Bangladesh, okay, Yusuf Akond in particular. And that gene is a single gene. I mean, it's a big result for a single gene. You don't get that very often. Back crossed into Bangladeshi varieties, grown in southern areas, very salt-affected fields in southern Bangladesh. There are actually 2 genes, I simplified this very good. But each one works by itself. In fact, if you put the 2 genes together, they don't work so well. If you put one or the other gene in, you're getting a 10 to 20%, often 20% yield increase in the fields in southern Bangladesh. This is in the boral season, the dry between the rice crops. It's a pretty good result. And so I think that's really quite a nice little story where you can do stuff that's completely academic sitting there, and then 20 years later, the results have been rolled out to the fields in southern Bangladesh. So that's a good story, that's an interesting story, but not so normal, I would say, in many ways. What I want to tell you now, we go to the next slide, is another story. That's just my calculations why the wheat farmers didn't told me to go away. Let's go to the next, and the next, and the next. Keep going forward. Right, okay. This is another story I'm going to tell you, which might be of interest. You've got a lot of tomatoes in Bangladesh. You've got a lot of melons, pumpkins, cucumbers. Right, okay. And a lot of this is done in open fields, right? Okay. So this is a story about tomatoes. And again, it starts academic. It starts weird and a bit useless. And I moved to Kaust, and in the first week I was in Kaust, a student walked into my office, she's called Evelyn, and said, could I work on salinity tolerance? Like, yeah, maybe. And she said, could I work on tomatoes? I know nothing about tomatoes. And so I remembered work. For some reason, my brain recalled a paper, a couple of papers, which published back in the 1970s, 1975, 1981, quite high-profile papers, where they were claiming to have tomatoes from the Galapagos Islands growing on these rocky volcanic rocks being splashed by the seawater, that these plants were having 100% survival when grown in full seawater. Whoa, 1975, nothing been done since those papers, all right? I went to the author, it's actually Manny Epstein, he's a very famous plant nutritionist, very respected scientist, UC Davis, and he said, he gave the seed to somebody else, and then they put it in a drawer, and there was a big story about what happened, and nothing happening after he retired. So I went, okay, do you mind if I take up this material? He said, all yours. It turned out, unfortunately, that these results have been falsified by the technician. He didn't know. So falsification in science has been going on for a long time. And that these plants actually did have the same type of mortality as normal tomatoes. But in the process, one variety, one tomato by an individual plant, in these big screens that my PhD student was doing, was very, very salt tolerant and was surviving. in these high salinities. This could only happen in an academic environment where you're sort of sitting there and then suddenly a new plant comes out. And it turns out that the plant was from a different species. It had been delivered to us by accident, by error. It's a classic of the scientific serendipity. At any rate, if we go on, what we've done is after 3 victims, PhD students, after 3 PhD students finishing their work, next slide please, and a whole bunch of research done by a range of people, even Bill Gates came and helped for a day at one stage. We had a lot of, the next slide, a lot of papers being published. This is the second victim, student. And then we started actually now my partner, Garby Feeder, who's on the beach side, started crossing these Galapagos and this other tomato with commercial tomato and getting halfway. We started thinking, right, we're going to have to get this salinity cost because these were big results. It's very, very spectacular results, even though we're doing the original Galapagos tomatoes. If we go to the next slide, you'll see that we did a lot of academic work, loads of papers published, that's all fine, but I still didn't know how to deliver it. So if we go to the next slide, what I realized, just it came to me, grapefruit dropped on my head, and I realized that what we would be able to do, potentially, because I'm not a breeder, I'm just a guy, a plant guy, what I thought we could do is deliver the outcomes of these pretty remarkable salinity tolerance results that we were getting more rapidly by just delivering rootstocks. And then that means we don't have to develop all of the breeding for the fruits. So it turned out that we were able to do grafting of these tomatoes. They were all compatible, everything was working, that was lucky. And then if you go to the next slide, we started to do plants where we were grafting straight onto the wild varieties directly. That led to a decrease in yields. The plants were salt tolerant, but they were weak because there was not enough vigor in those wild varieties that are tough but not vigorous. So then, of course, I remember that most, in fact, all commercial rootstocks in the tomato world are F1 hybrids. F1 hybrids between the wild species and the commercial, always developed for increasing disease resistance. And I was trying to increase abiotic stress tolerance. So it's pretty similar principle. So then I thought, okay, let's see if we can emulate these very, very successful commercial varieties by making F1 hybrids combine the vigor of domesticated tomato with the salinity tolerance of these wild tomatoes. So if you go on to the next slide, what we did was actually it ended up being a pretty big research program, lots of people involved, and yes, the third victim, PhD student, slave, whatever, doing this work, and we ended up crossing, it was I think over 60 different wild varieties of tomato, accessions of tomatoes from half a dozen different species, and we did some big screenings in greenhouse and field, and ended up with some hybrids which do seem to be quite distinct and very successful. So if we go to the next slide, you should see, yes. So what we started to do was scale this up to field trials in multiple countries around the world. This is now flipped over, by the way, I should have pointed out. I think this is the first slide, to my company. So we started up a company because we thought we were onto something. And you can see here, this is in a greenhouse of the actually largest field tomato processing tomato company in the world. And they grabbed hold of this. They love our rootstocks, and that's a green, there's a third one. Actually, they're now grafting millions of these plants with our rootstocks, which is pretty interesting. So that's been grown in California. We've got farms in Mexico, Spain, Portugal, Egypt, really good in Egypt too. So if we go to the next slide, what we find is that lots of the results are very, very positive. This is just some examples. And you might look at that number over there and say 26% yield increase. All right, that's pretty good. But you've got to look at something else as well. This is with half of the planting density. All right, or less than half the planting density. Because these rootstocks make the plants grow like they're on steroids. You wouldn't believe it. These great big giant plants. And so they now actually not only have to grow them at half the density. They want to grow them at half the density because the input costs plummet. So you end up with something which is complete game changing in open field horticulture at the moment, which is, I'm proud to say, come from our work. Because you're going, okay, you don't graft in open fields. You should have seen the response when I went to the head of the Florida Tomato Growers Association. I said, oh, we've got this plant, we're getting some really interesting results, trying to give it a trial. And so I described a little bit, and he says, no, we don't graft here in Florida. I said, no, I know you don't graft. Like, there's no grafting of tomatoes in open fields. You just don't get the returns. And so I said, no, I know you don't graft, but we're talking about more than half of your inputs. You've got the cost of the grafting, which is 2 seeds rather than 1, and the cost of the grafting, so that input cost is higher, but you're planting less than half the number of plants, so you're already ahead. You've got less water use, less fertilizer use, and a 25% increase in yield. And he said, no, you're not listening to me. We don't graft in Florida, all right? Now get away. That was the Florida people. They told me to get back in my box. The Californians went, this is cool. The Mexicans love it. They're grabbing it. There's a lot of people around the world now taking hold of these tomatoes because of these remarkable decreases in input costs that are more than offsetting the increase in costs of getting that seedling because you need to graft. In California, you might go, how do they do this? Because of high labor costs, and the reality is now, letting out one of our big secrets, but it's robotic grafting now. So the company's got 6 robot grafting machines. They can turn over millions of seedlings they can generate each season. I didn't know if it's millions or tens of millions, but I think it's millions of seedlings. Now I know for tomatoes in Central California each year it's 1 billion plants being planted. In Egypt it's 1 billion plants being planted each season. It's a lot of plants. But, so we're still down in the millions, but it's not bad. It's heading in the right direction. Okay, next slide. Oh, now this is important, of course. You've got to make a business case of fun and not just wave your arms around and say, hey, 25% yield increase and half the number of plants per hectare. You've got to do your math. And what we found is in multiple field trials now, in multiple locations, but especially interestingly in California, what we're finding is that you're increasing yield with half the density of plants. You're increasing your profit in the order of $3,000 per hectare. This is pretty serious. That's a lot of money per hectare. That's about a 50% increase in profitability. So I think we're looking at quite a significant, interesting development where the system has never been grafted, you're now able to graft. Okay, if we go to the next slide. All right, that's just more results. The fact that this all originated with salinity tolerance, and yes, the plants are more salt tolerant. That's probably beneficial, but of course you need lots of other benefits as well. Especially with climate change in places like California, reduced irrigation demands. Okay, these roots are crazy. They look like they're on steroids. They're so big and strong. Now, if we go to the next slide. Okay, so I was doing some experiments with this guy, a professor at Kafr Sheikh University, Mohammed Raqqa, and we're in this field. You can see it was setting sun, it was beautiful. You could see my plants above all the others and heavy with fruits. And I just stood there on the edge of the field and I said to Mohammed, why do you guys graft in? This is such a standout result. And he said, the seed is too expensive. Not the grafting process, the seed is too expensive. I said, well, what do you mean? He says, what? It's 20 cents for these rootstock seeds. And they're F1 hybrids and blah, blah, and then you've got to put another F1 hybrid on top. The farm just doesn't get enough dollars back per plant for this investment to be worth it. And so that's when I had my second, this time it was a watermelon, I think, landing on my head. What I realized was if we could reduce the cost of seed production, this could be quite important. And so what I did, if you go to the next slide, was I realized that we could tetraploidize the interspecific F1 hybrids. Why not? Why not? Because every tomato grown on the planet is diploid. There's been 100 papers where people have tetraploidized tomatoes and failed, but they're always doing it as an intraspecifics. These were auto-tetraploids. Okay, they might have done wide intraspecific hybridizations and failing, but this is a wide interspecific hybridization. So there's much more possibilities for these synthetic new allotetraploids, interspecific allotetraploids, to actually work. So there was a bit of a gamble again. I think the university could do this. It'd be hard for a company to take this on, but we did it, and it worked. All right. So we've now managed to get plants. They look okay. It's a new species. You do these interspecific allotetraploids. The plants look completely different. They're self-compatible. Thank goodness, I had a lot of luck. All right. They seem to, they're not always stable. All right. And that's, we've got a research project, victim number 4, it's student number 4 coming along, who's trying to work out the genomic or genetic, we don't know what, basis for some of the lines not being stable. So it's not perfect, I will admit that. But I tell you, 80% of the time it's looking like it is working. So we allotetraploidize the F1 hybrids, and if we go to the next slide, we have to patent it, of course, because it seems not to have been done. And I'll tell you the final icing on the cake in a minute. But we did these allotetraploids. If we go to the next slide, please. You're getting much cheaper seed production. All right. Now I know that also means you can't protect your seeds as a normal F1 hybrid, and that's a company business decision that we have to make when we're going to release these and to whom. And you want systems. So if you're in Central California, it's probably okay. In other places, a bit more Wild West, it might be a bit more difficult. But I can tell you there's a lot of reasons to go with this. And if you've got this greatly reduced seed cost, and remember we're doing rootstocks. You can't eat these things. The plants do come up, they fruit, but and they taste awesome. Actually, they're a deep red color, the fruits, but they're full of seeds and they've got a thick, tough skin. There'll be a whole breeding program to turn them into something that the consumer would want to eat, or the processor would want to process as well instead. But at the moment, seed costs massively reduced, so we're way below 1 cent production per seed. So you can still sell them, and there's all sorts of business things which we can talk about in a while. But what's really interesting is this next step here, where I'm calling this the F1 text. So I was walking through a greenhouse of a seed producer, because we're producing seed commercially. We know those millions, tens of millions of seeds, you have to. And I was going through one of these production facilities, and then the guy, he was the managing director of the company, was walking me through the greenhouse and says, "Hey, do you know what these plants are?" And he sort of just hit it, and I could smell, and I said, "Oh, is that Panchobrihi?" So I passed the test, because it was this language sort of smelly, wild relative of tomato. And he said, yeah. Every rootstock we made's got those in it. And he said, "Oh, that's a pain to grow these plants. They're difficult to grow, bad pollen, but every rootstock that we have to generate seeds for has that in it." And then you can see him sort of freeze and blush. And he should have said that. He should have said that. And then I was freezing and blushing because I didn't have that in my rootstock. All right. So what's going wrong? What have I done wrong? I was just petrified when he said this. He was petrified because it released his trade secret. I was petrified because I didn't have this species as one of the parents in my lines. And I'm going all the way through F1 hybrids, selling millions of seeds, tetraploids. We're not selling seeds yet, we're very close. A lot of field trials still. But what I realized is when I had the third, this time it was an apple, land on my head, was if we've got a tetraploid that's got my original wild species in it with a commercial, it doesn't have that Panchobrihi in it. What I can do is make a tetraploid from a F1 hybrid that's got the Panchobrihi, think some of you are already with me. So we've got that tetraploid and this tetraploid, then we hybridize the tetraploids. Nobody's ever done this. So that was another component in my patent, which has been published. This is what I can talk about. And so we've got now tetraploids, interspecific other tetraploids, and then hybridizing. So what you're doing is you're stacking 4 different genomes into one bar. It's beautiful. It's absolutely beautiful. So you can stack abiotic stress tolerance, you can stack biotic stress tolerances. We've even got Orobanki resistance, all right. So, you know, and you look at these are often ones which are multigenic. You need the whole genome, all right? And so you can put in the whole genome, not just the gene. And so the only problem is if your trait is recessive. All right, so if your trait is recessive, then it's not going to work. But I can tell you a lot of traits are not recessive, a lot of them are dominant, and so it's working very beautifully. And of course, a lot of them are dominant. You all know this because half of you in the room work on F1 hybrid rice or F1 hybrid pumpkins or whatever it is you're working on. All right, so if you take in disease resistance in, that's dominant. All right, that trait in your F1 hybrid. So we're sort of making what I call lines which have got super heterosis. All right, got 4 different genomes combined, and you're also enjoying the vigor of polyploidy. Right, so you're getting polyploid vigor and super heterosis, and the rapid stacking effects. You can do this whole process from wild to an F1 hybrid in 2.5 years. That's breeding on steroids. Okay, this is how the roots look like. All right, these are different examples. This is from from my very first field trial in Egypt. I still use this picture. It was in a shallow sandy soil with the clay subsoil. It's very saline. It's a nightmare site. Those are the normal tomato roots. Our roots just they punch down through into the clay substrate. They're probably eating the small soil. But anyway, that's a really, really nice, incredible. And you dig up the plants, it's ridiculous, the difference in the roots. Thank you. So, yeah, I think that's probably enough. You're getting big yield increases. The field trials now are nowhere near as extensive as our F1 hybrids, but that's okay. We're getting there. This year we've really gone crazy, and we're on every continent with over a dozen field trials. With like over 1 million seeds. Okay, yes. We're not just doing the tomato, of course. So you start to do that. I did have beginners like I know, but I'm going to build on that and try to grab it. We're doing with eggplant, which has been working. Trying with pepper hasn't been working yet. We've made allotetraploid grapevine rootstocks, so go figure. That's going to be quite interesting. We've also done it with walnuts. Melons, oh, this is tricky. So we're making squash, interspecific squash rootstocks that are allotetraploid. These are working, but we're still in the early days. So I can't go around trumpeting that. But if we really do manage to get thinking permutation also working, I think that will be a happy day. And that's probably enough. We can go on to the next slide. And this is how we started the company. So it's a company which was founded with an engineer, and we started it growing tomatoes in saltwater, seawater cooled greenhouses. So we're trying to develop a saltwater, seawater-based agricultural system. I don't think that's working yet. I think we've still got a fair way to really get all the way through to a full seawater greenhouse. But in the process, we made a lot of noise, we attracted a lot of attention, and we attracted the attention of this young Turkish professor, Derya Baran, who's a material scientist, and has generated a heat-absorbing nanoparticle, which you can disperse in plastics, and it reduces the heat load, the infrared. It has pretty close to zero absorbance in the visible, and it has a substantial absorption in the infrared. So you get less soil heating or less substrate heating. You're getting a greater ability to control the temperature for longer into the day. You don't have to put the shades across as early. You don't have to use as much water for both the cooling and the irrigation. So you save on electricity costs, you save on water, you get higher yields. In fact, what you have to do, you have to teach the farmers, because these are being installed as many, many square kilometers greenhouse with her heat blocking nanoparticles on now. In fact, she's making a profit, I'm still making a loss. So this is a great merger when her little spin out merged with my company. And it was one of the better deals I did in my life. But what you have to do is teach the farmers to water less, especially in the cucurbitaceae. So when we first did the first cucumbers, the farmer came back and said, "This is rubbish, my yield's gone down. You're overwatering." And he was overwatering his plant. So it's so effective, that heat blocking. It's beautiful. Okay, so that's how our company started. We started in production, no way. We pivoted very quickly to being simply a technology play. So if we go to the next slide, we've actually built it up so we've got a lot of manufacturing partners now in multiple countries. So you can click on catalogs now, go to Armando Alvarez, they were our very first partner, massive plastics company, and it's on the catalogs now, so you can just buy it if you want the RS heat blocking. And yeah, we're still doing a bit of demonstrations and so on, but we're now starting to crank it up in the Middle East, and we've now got sales in 15 countries on every continent. And yeah. Okay, so that's how the company started. So we've got plastics and rootstocks. The plastics are delivered very rapidly and are I said profitable earlier. I think they actually are now. And then we're still doing our tomato work. Thank you. And I think that's enough. Okay, that's just for the students, so we can go over that. You guys don't do money, we can go over that, that's how to deliver. That's just my last slide, staying with it, what to do more. Thank you all very much. Too long, sorry.
Thank you. So now, Mark and Abed will take questions. So I invite both of them to go to the stairs again and sit in the chairs.
Oh, chairs.
Yeah.
Thank you.
Or just Seva also please join there because we have this similar thing.
Share ourselves.
No, okay, somebody will join them. Okay, if point about the so from ACI, answer is high.
Question over there already.
Yeah. Do we have any questions?
There's a question over here already.
Yeah.
Do you need another microphone?
Thank you, Professor Abed and Professor Dr. Mark. I have two questions, one each of you. First one, I would like to ask Professor Abed Chaudhury. You showed that Panchobrihi lies in the Panchobrihi. Panchobrihi lies in the huge number of groups. What's the factors involved here to induce huge number of groups in Panchobrihi crops compared to transplanted crops?
Hello. Is it working?
Yeah, it is okay.
Yeah, so multi-harvest rice, you can say Panchobrihi, sometimes people don't understand Bangla. It's very, it's maybe better just to say multi-harvest as opposed to mono-harvest. So mono-harvest rice has been selected in during crop domestication because this rice was multi-harvest perennial. like 10,000 years ago, and then domesticating farmers selected seasonal rice out of the perennial crops. And any perennial crops like sweetgrass, Lonium perenni, they have massive root system. So perenniality and root system goes hand in hand. We selected seasonal rice, bull rice, house rice, almond rice. And in the process, we destroyed the root system. We destroyed, like wheat is a good example. Modern wheat has a TIM mutation, TIM, which suppresses artillery. A lot of modern rice has been selected to not to have artillery. So this is why they also have less root, because no one selected to have less root. But when they selected suppresses theory, they wiped out root without realizing it. Because who digs up the root? Who digs up the rice and looks at the root? It has never been done by anybody. So when I started to do multi-harvest and selection for multi-harvest trade, and only multi-harvest about ground trade, I realized much later that I created rice with massive root system, 50 times more root. Root is so big that my farmers have to dig like a huge hole simply to get them out and destruct them. So I think it's a part of the domesticating selection and subsequent. We can call it is perennial because we have a very harsh winter. So when I nursed some of them and kept them in one condition, they seem to have become gone into the second year. So I can't say, yeah.
So do you do any softening of the soil so the root system goes?
No. Well, it's pretty wet condition in the village, right? I mean, it's not hard soil. But we don't do anything deliberately like making it. No, this root, it's unreal actually, unless you see it. And for the longest period, I didn't even know. After 7 or 8 years, I realized. And plants seem to be going foraging deep into the soil. Different, it's heavily dependent on the density also of planting. Also, so they kind of get all tangled and created continuous an ecosystem under the soil.
Yeah, it's a relevant question. Just to use the class this question. What was the process of your modern selection? You talked about 2 types of selection.
Yeah, so I had to know a lot about the rice currents, aquatic groups, very large number of rice coming from different ecosystems. You have to realize that I've been doing it for 24 years, right? So I first collected all conceivable rice varieties that I could get my hand in, and then looked into the generic properties, and then cross one with the other, and then that cross with the another one in different aquatic groups. So it's a very long, elaborate breeding system which gave rise to multi-harvest.
One question to Dr. Mark. Yeah. In one slide, you showed that the density of tomato population goes down and then yield increase, right?
Yeah.
So do you think the only density is a factor for increasing the yield or some other factors also involved here?
Okay, no, so what's happening is a very controlled system. So the growers are deliberately choosing to plant at half the density because the plants are so immense. So the root stock adds so much vigor to the shoots that you actually have to plant at half the density. So it's definitely the graft, the root that is causing the need to grow at half the density.
Thanks, sir. So I have a question for Panchobrihi. Sir, you mentioned that you develop the online fabric technology or automatic technology to develop in Panchobrihi. Is it possible to use any inbreed line or explorer line or material line, but is functioning male part and is the female part?
Yeah, so these are 2 different projects which I happen to have done. So in the 90s, I started the Prometheus project, and that led to finally doing it in rice. So that's one project that I did with CSRO and PV collaboration. Panchobrihi multi-harvest is something I did along with the farmers. So these are two different ones. But it occurred to me that getting rice perennial is one way of doing it economically, without going into the seed part, but simply make it vegetative. Get the same result. So imagine if you have a multi-harvest hybrid rice or a perennialized hybrid rice. Then the farmer just buys the hybrid seed and then maintains it by like Napier grass perennial. Most people find it so unhappy, kind of hostile in a way from the seed company point of view. They don't want to accept it. Then you will just buy hybrid seed and that's it, and then you have it. So it's not very good for business. Right? So then people say it's not going to happen. But rice does have the inherent ability. And if you look into horizontal supercon or horizontal Ibarra, so Jagiburo is horizontal Ibarra. Jagiburo is still being grown in Sunamgan power. And Jagiburo still remains fairly perennial. So it's not so strange.
But seed companies still have designs because they're going to, they still have to be seed control.
No, that's another discussion that I could have with Dr. Ansari. I'm not actually familiar with all the business aspects of it, but I simply like do and let nature take me wherever I go, right? So I don't put a brake on it. But at some point we have to say whether it's business, whether it can be developed by business model or how and why. But it has occurred to me, many people pointed out to me that we need to have a discussion with the people who are making money by selling hybrid seeds to see what is the implication of what they have as hybrid seeds. You introduce yourself. Can you say APK Boremek too?
Dr. Mohammed, Principal Scientist, ACI Molecular Science. Actually, I don't know, have you done any care modification in your Panchobrihi?
I have done it by forward breeding, which means I deployed the parents and selected the trait and take it to about F10 or something. And I have about 200 varieties where the parents are different. So I am assuming that I have messed around with a large number of genes. And now I have to backcross it with a non-multi-harvest and get an F2 parental population and map the genes. I have not done it. And then those genes will be the target for one-step CRISPR to turn any rice into multi-harvest. So this is the strategy. I have simply developed, you can imagine, 200 independently isolated mutations. I would think it's a fair chance that I have gotten a large number of genes responsible in this trait, and many of them would probably be common. But now I want to talk to Dr. Ansari and people like that and launch a massive breeding program to fish out, ferret out all the genes. So in future, like in next decade or next 5 years, those genes will become targets for CRISPR.
Thanks.
I have a question. I'm not a vice scientist. I am a retired executive director of corporate development. Now I am working in the Supreme System Party as a quality member. I'm mostly dealing with whatever. But out of my curiosity, the multi-harvest price, do you have any starting on it's the monoculture? The pressure, waste and disease pressure will increase if you keep the same price in the field. And another one is, do you just recommend it for any specific ecosystems? Because we have defined land types and rice contribution is not possible around the year in the however area.
So the answer is there has not been to my, because I'm doing it for about 10 years now. Once we have the multi-harvest rice, I've been growing and developing new varieties. I have not noticed any disease pressure. If anything, these varieties get very strong because of their massive root system. They seem to be different from ordinary rice. So I don't accept, I know it's in the textbook, it says that if you keep rice growing, it will have disease pressure, but at a qualitative level, I have not seen it. Number two is that this is not compatible with the our ecosystem, because you can't at 35 centimeter height, and if the water comes at above 35 centimeters, then you are not going to have much more. So you need to have non-our ecosystem.
I have a question. Do you have any plan to make it popular variety? So what is your plan to make it?
Of course. It is spreading one village by one village. We have a 7,000 village, and I have covered maybe about 20 villages. So it's like a biological fungal growth. It is going to spread through the country, village after village, as a farmer-led innovation.
Yes, that is fine, but if you keep your field occupied by only one crop, then there is a threat.
Yes.
Of producing other yields, either like vegetables or other crops.
Yes.
So what is it in that case?
Yes. So farmers don't have the obligation to do five harvest. They can do a burrow and a house. It actually does very beautifully burrow and 2 consecutive hours in 100 days. So imagine they do that, and then after they already got 3 harvests, and then rest of the time they can do other vegetables. So there are all these options, and we have simply creating the competence. We are not making it mandatory that people must do 5 harvests.
Okay, thank you.
Assalamualaikum. I'm Dr. Sultana from Lab Limited. Sir, with due respect, I would like to mention one thing. Is the multi-cultivated rice, the Panchobrihi rice, is too deep root system. If one farmer wants to cultivate another crop next year, the root system doesn't make any problem for the farmer.
Root system, whether it will create mechanical problem or not, because after going taking the tractor in there, we need to do simulation experiment. But one thing I can say that because of the massive root system, the soil carbon goes up to about 3%. And normally soil carbon is less than 1% in my village, in random farmer's field, but it goes up to 3%. Now what you are saying could be true, that there is a massive number of mechanical, like root debris, and how that will impact the next harvest. Well, in my case, when I plant rice again, it doesn't, but if you want to do potato, yeah, I think we have to do this kind of thing on a case-by-case basis and get real data rather than, I'm not wise enough to suggest some outcome. Yeah, feedback from farmers, do some simulation experiments, because this is creating a new agronomy, so I don't think I have all the answers.
Thank you, sir. I'm Dr. Abdul Qadir, Chief Scientific Officer, Bangladesh Rice Research Institute. Especially, I'm working on nutritional rice, including transgenic rice, as well as restaurant rice. So my first question is to, I have some question actually. First question is to Dr. Chaudhury. The question is, you mentioned the 110 days needed for first generation or first cycle. So what is the starting point? It is starting point of the rice. Is it Hausamam or Burro?
We start with burrow at the beginning of the year.
Yeah, okay. So if it is Burro, starting point is Burro, I don't know how it is possible to harvest within 110 days.
Because we get super
This is the question. And then another question is, you mentioned clonal or atomic systems. I don't know what is right concept. Is it clonal or atomic system?
Clonal simply means.
You mentioned in your speech.
No, but let me answer. Clonal simply, clonal is a generic word, right? Apomixis is a very specific technical term. These are two different terms.
Yeah, okay.
So clonal means, when you say something is a clonal copy, it means that it's identical to the market. Right? It doesn't segregate.
Yes.
That's what clonal means.
All the vegetative propagation is like it.
No, yes. Apomixis definition is that you deliver clonal outcome through seed.
Yeah.
And by vegetative body, you deliver normal outcome to vegetative body. Genetic outcome is the same, whether you do it by the seed or you do it by the vegetative body, it's the same thing. So I use it interchangeably. Some people just don't know what apomixis means. So to them I say clone.
Okay. The question is, you want to develop apomixis seed, but need seed. In case of apomixis, it needs seed. It is not true seed, it is not fertilized seed, but seed. Is it correct?
Do you know that there are many organisms, hundreds of organisms which are apomixis?
No, in case of rice, I mentioned about the rice. So in case of rice, you develop apomixis seed or true seed. But in both cases, if you want to cultivate rice, you need either apomixis seed or true seed. Am I right?
I don't know what your question is. You're making some sense.
My question is, why we are going to the develop apomixis seed without pollination? What is the benefit of apomixis seed than true seed or pollination?
The benefit obviously is the elephant in the room. The benefit is that it propagates hybrid vigor without segregation forever. That is how apomixis is defined, right? Hybrid seed will give children with a hybrid seed. You understand?
Okay.
আপনি এটা বুঝতে পেরেছেন? শুনেন, hybrid seed আপনি একটা প্রশ্ন করতেছেন, তো এই যে ডক্টর আনসারি হাইব্রিড সিড কেন লাগে? কারণ দুটো প্যারেন্ট অনেক কষ্ট। থ্রি লাইন হাইব্রিড একদম মাজা ভেঙে যায়, তাই না? একটা ক্রস করবেন, ইন্ডিভিজুয়াল লাইন এটাকে অ্যামপ্লিফাই করে আরেকটা লাইন করবেন, তারপরে আপনি হাইব্রিড বানাবেন। আপনি হাইব্রিড বিক্রি করবেন 500 টাকা, তাই না? ফার্মার এটা কিনবে, যখন ফার্মার ধান কাটবে তার সব ভেঙে গেছে হাইব্রিড, ওই সিডটা লাগাতে পারবে। আবার আসবে এসিআই কাছে, লাস্টের কাছে, আবার দাও, কিরা দাও, এগুলো দাও, মুক্তি নাম আছে, আসুকায় আছে, কিরা, তো এগুলো করবে। তো এখন হবে কি, আমি বলি আমার কনস্ট্রাক্টরে যখন আমি হীরাতে ঢুকাই দেবো, তখন হীরার বাচ্চা আবার হীরার হবে, সেগ্রেগেশন হবে। এই কারণে এটা হয়, এটা ওয়ান অফ দ্য বিগেস্ট ফ্রন্টিয়ার ইন প্লান্ট ব্রিডিং ইজ apomixis.
Okay. So next question is, I don't know you two technology. One is Panchobrihi rice and other is apomixis. This is different. So two technology. If it is mature, I think it will go through the national system. It should be national system. Otherwise, this technology is very difficult to go far.
difficulty নিয়ে আমার কোনো অপোজিশন নাই। আমি একজন ইন্টারন্যাশনাল মানুষ, এই দেশ না করলে কম্বোডিয়া করবে, বার্সেলোনা করবে। এই দেশ যদি করতে না চায়, আমি তো কাউকে করতে বাধ্য করবো না, তাই না? এটা অন্য ব্যাপার, ওই আলোচনা এখানে নাই করবো। সরকার কি করবে না করবে আর আপনাদের সিস্টেম কি করবে, এটা নিয়ে আমার কোনো বক্তব্য নেই। আমি সিম্পলি জেনারেট দ্য টেকনোলজি, গিভ দ্য অপশন, তাই না? আমি একটা গান লিখে দিব, মানুষকে গাইবে নেবে গাইবে, এই হচ্ছে ওকে। আমি লিমিট নাই স্যার, আমি কোনো কাউকে গান গাইতে পারবো না।
Another question to Dr. Mark. NAXG, you mentioned in your slide no yield penalty by increasing salinity by introducing NAXG. First question is it a genomatic product or a transgenic product?
I didn't tell the story. It's a good question. It was actually originally, so the original wheat, from the original hybridization, tetraploidization of wheat 10,000 years ago, the genome donor happened to not have that gene present. And the gene was brought in from another accession of the big genome donors that are out there in germplasm collections through sexual hybridization. So it was a non-GM sexual hybridization which was bringing back in to, it's a classic genetic bottleneck, breeding domestication story. So It just happened that the original durum wheat had the bad luck of not having an exon gene in its genome. It's just physically missing in the genome. And other wild relatives, diploid wild relatives, did have that gene. So it was just introduced by hybridization.
Okay, what level of salinity did you perform? What level of salinity? No penalty of it.
Yeah. So the numbers actually are in the papers. What we're talking about, this is a dry land system that we're working in Australia. And so the soil salinities are not really too useful. What I'm talking about, in Australia, we had fields where we had 3 tons, in fact, we even had 4 tons per hectare, and we had 1.5 tons per hectare, so the yield had been halved by the wheat because of the salinity in the soil. Then that gene was bringing back 25% increase in yield.
The gene is transferred to rice. Is it possible or any option to transfer the gene in rice?
Yeah, rice has the gene. It's on chromosome 1 in rice, and it's a very closely related gene. Rice is an interesting problem, which actually probably isn't related to its ability or lack thereof to exclude sodium from the shoot. Rice seems to have has a shoot which is very sensitive to the accumulation of rice in the shoot, which is different to wheat and barley. So that what we call the tissue tolerance of rice in the leaf is very, very low. So rice does have HKT1. There is a little variation. It is related to the extent of salt accumulation in the shoot. That's already very well established. But I think there are other components that are more important in rice. Rice is very salt sensitive, and it actually doesn't accumulate that high a level of salt in the shoot. It's just a weak and pathetic shoot. All right?
Okay, thank you very much. Thank you.
Three questions for Dr. Mark. So you mentioned tetraploidization for your rootstock. One, it's making the sturdy roots, I understand that, and of course, sturdy roots are very important for picking up nutrition or whatever hazards that the soil has. You mentioned tetraploidization for increase in seed. How does that work? That's one question.
Yes, okay. So what I'm talking about is sort of similar to the apomixis, where you're just producing the seed without meiosis. Here, you still produce the seed with meiosis, but the tetraploidization makes the plant homozygous, so it's going to breed true. That's all. So what I say is it's immortalizing the hybridization.
In relation to the big bird. So the bird has a lot of meiotic events and reduces seed set. When you tetraploidize, all those non-viable seeds disappear, and suddenly you have a massive boost. So all allotetraploidization, for instance, this is happening during domestication. Wheat is a good example. So wheat swallowed up so many genomes, A, B, D genome, and each genome is being deploidized to an extent, leading to hexaploid wheat. So during the process of hexaploid formation, there are a lot of non-viable intermediate proteins which got fixed by polyploidization. And for wheat, hexaploidization was the target outcome, which delivered a massive crop.
Second question, Mark, that is, you mentioned that you want to take this technology to eggplant, which is partially sugar, and others. So for each of these, you're going to take wild varieties of, say, eggplant or others, and try to tetraploidize it and use the same exact. That's the. I mean, you can't use the tomato rootstock to graft like things which are closely related. No.
No, you can do things like put tomato on eggplants, which we've actually done. We've been collaborating with the wild eggplant that actually grows quite spread across South Asia, called Solanus siciliifolium. So it's actually, our head has been doing some of that work in Silet.
Okay.
But But generally speaking, to maximize graft compatibility, you really need tomato on tomato with eggplant and eggplant.
Sorry, I have so many questions. So the NACs, you mentioned that in the wheat, in Bangladesh particularly, either it's NACs 1 or NACs 2, but when you put both of them together, they're not working that well. That's one question. And the question is, has it been released to the farmers, the wheat in Bangladesh?
Yes.
That's one. Oh, it has?
Yes, I understand it's being released by Bari in southern Bangladesh. And I don't know the acreage.
Okay.
But I understand that it has been released.
So Dr. Yusuf Ahmed would know the details.
Exactly.
Okay.
Exactly.
Great. So
and the next one, next two question, that's just, I just say it's too much of a good thing. All right.
But it's different, right? I mean, it's like 1-4, which
1-4 and 1-5.
1-5. So 1-5 is, we discussed the
yes,
challenges this morning. The 1-4 is it sequesters the salt in the older leaves, as far as I understand.
Yes, it's in the base of the shoot, so you may have an effective letting a bit of salt.
It should be complementary, right?
Yes, it should be, but I think what you actually might have, especially in moderate saline soils, where the benefits are pretty low anyway for these genes, is you're actually inhibiting growth, because the plant needs a bit of salt up there for osmotic potential and for expanding cells and so on.
So my last question is, you mentioned, you kept on saying plastic for absorbing heat. Can you explain that a bit more? Because
yes. Okay. So I mean, I'm very happy with this. I'm very happy with Daria Baran, Professor Daria Baran, material scientist. And so this is a classic where you have different fields coming together and you really do get synergies. So she's a material scientist. She was actually working in a solar center to try to generate nanoparticles that were absorbed in the infrared and separate charge. So she was trying to develop an infrared absorbing solar panel. And she was successful at doing that. And then she brought the nanoparticles to us and said, could I put this on the roof of the greenhouses? And so we went, sure. But then you look at the economics of this, it doesn't work. I mean, it's way too expensive and it's not viable yet. Okay, where our company owns the IP for these infrared solar panels, but we're talking years away and we just don't have the capital to do it. We're still a mid-sized company. And in the meantime, it was actually Ryan, my original co-founder, who's an engineer, he said, forget solar panels, let's just disperse these nanoparticles into plastics, because they can just be a heat blocking compound in their own right, full stop. Forget all the fancy solar panel stuff, and the nanoparticles themselves, so the basic raw material, is really cheap. So you can disperse this into plastics. You can't put them on glass because you can't disperse them into glass. But 95% of greenhouses on the planet are plastic. And even better, those plastic roofs get replaced every 3 to 5 to 10 years, depending on the plastic. And so funds are always replacing them. So what they're doing now is, we tell them about our heat blocking, and they say, well, I've got to replace 10 hectares. I'll put your stuff on 1 hectare and see how it goes. So that they test it themselves. It's just common sense.
It doesn't block the light.
It does not block the light. It's called Second Sky. That's our marketing name because it makes the plastic look blue. And I always remember when I first saw one of these bits of plastic, I said, that's blue. You said it doesn't absorb any visible light. It's like you can imagine it turning aggressively straight onto Derek, actually. So you said it doesn't absorb any red light, and this is blue. And then she actually had to explain to me that it's not because it's absorbing red light, it's because it's scattering the red light, exactly the same as why the sky is blue. And so that's why we call it second sky, because we're now making second sky over the agricultural crops.
Sorry, a supplementary question, because it's, how long does it last? Does the...
Yep, the nanoparticles last longer than plastic. We've
Okay.
had like immeasurable decreases in its effectiveness.
So how are you
Not in
doing
the
this sustainably? I mean, the plastic.
Oh, well, it's good you ask that because our fundamental drive is to reduce the environmental footprint of our food production. And we've now gone into partnership with SABIC, so a very big chemicals company. They have developed a recyclable polyethylene plastic. And I think it's real. I don't think it's just marketing spin. And so they've put our nanoparticles into their plastic. And so then it gets it goes into the recycling process. Now look, you saw we've got a dozen manufacturing partners now. It's certainly not only in recyclable plastic. I was fantasizing that our nanoparticles might increase the lifespan of the plastic.
Yes,
Wouldn't that be wonderful? We've got no evidence for that, unfortunately.
Just one question, two questions for Dr. Chaudhury from online. So, Mr. Salimullah, he asked one question that how does the Panchobrihi performance fare in terms of yield, cost, etc. The other one is that whether it is genetically different from the normal variety. So these are two questions from Mr. Salimullah online.
So yield is, it starts with about 6 tons and then 4 tons. Sometimes it bounces back. Some varieties that are yield is very high. So,